So what we are going to cover in this video is the process of DNA replication. That's a very important and little complex process as compared to the other ones that we've already done. So we are going to explain the replication of DNA in terms of unwinding of the double helix and the separation of the strands by helicase, and then the formation of new complementary strands. So basic three processes would be there. First would be unwinding of the double helix. The second process is separation of strands by helicase. And the third process is formation of new complementary strands by DNA polymerase. So before we start going through the processes, a few important things about the two enzymes. Over here in this process we need helicase, and in this process we need DNA polymerase. So what do these enzymes do? Now helicase, it unwinds the DNA and it separates the two nucleotide chains by breaking the hydrogen bonds between the complementary base pairs. And the two separated strands, which have been broken due to breakage of their hydrogen bonds, they act as templates for the synthesis of new strands. So that is what helicase does. What is the role of DNA polymerase? DNA polymerase, it synthesizes new strands from the two parental template strands. So the parent DNA would be split up into two, and from each of those two parent strands, new strands will be made by DNA polymerase. What actually happens is free deoxynucleoside triphosphates, deoxynucleoside triphosphates. These are nothing but phosphates with, these are nucleotides with 3 phosphate groups. That's why we call them 3 phosphates, triphosphates. So these free deoxynucleoside triphosphates, they are aligned opposite their base partner, and a covalent bond is formed. And the energy for this reaction comes from the cleavage of 2 extra phosphate groups. So the energy comes from the cleavage of 2 extra phosphate groups. So this part is important. So there are 3 phosphate groups, out of which only 1 will be used up, and the remaining 2 will be used to provide energy. And now you need to know what is the significance of complementary base pairing in conserving the base sequence of DNA. Now you know each of the nitrogenous bases can pair only with its complementary partner. That means A with T and G with C. So when DNA is replicated by the combined action of helicase and D polymerase, what all happens? The new strands will be identical to the parental strands or the template strands. Secondly, the 2 DNA molecules formed will be identical to the original molecule. So not only are the strands similar, the molecule as well is completely identical. Now DNA replication, it's a semi-conservative process. So let me draw a diagram for that. So let us suppose that this was the original DNA strand. Welcome to this video by split up into two portions on biology. Now a new DNA will the structure of the DNA, and we've also done the process of DNA replication. And what we're going to do in this video is transcription, and there were pairs between them, two bonds, or so once again, welcome to this video. Similar kinds of bonds will be created here. And this is Preetinder Kaur from Perfect Scores, and now so that is how started the process with the processes of transcription and translation. So we can say that it is a semi-conservative so process. You need to be focused and you need to concentrate a lot to understand it. Whenever a new double-stranded happens in the molecule is transcription. One strand transcription from the original process by which one will be RNA sequence. So there's no one that you can see over here. This is the new one. The yellow one is original. So that is why a semi-DNA template process is followed. And I hope so that was all that remember what were the differences between DNA and RNA. The next important thing that you need to know is the difference between the DNA and so coming back to transcription, an RNA DNA is deoxyribonucleic from a DNA template, and so RNA what are the different processes involved in nucleic acid. First of all, there is an enzyme so in DNA known the sugar as RNA deoxyribose. And in RNA, the sugar is just ribose. In DNA, the pairs or the nitrogen helicase does the same thing in DNA replication. But in this case, RNA, there is one change. Instead of thymine, we have uracil and synthesize. So thymine in complementary is replaced by uracil of RNA. DNA is double-stranded, so two strands work to form a helix. RNA complementary is usually single-stranded. One of the separated not DNA always, strands. But DNA is always double-stranded. So DNA will always form a double helix. And how does this happen? This happens by covalent. RNA does not form a double helix. Bonding the ribonucleoside. So that is all that we will be covering in this video. And in the next video, a very important process will be covered, that is transcription, that align opposite, and translation. So don't forget to watch that video to their exposed complementary partner. And don't forget to visit our website, perfect-scores.com, and share and like us when a DNA strand is left alone without its partner strand when it is separated at gmail.com. The complementary, the base pairs, they are much for watching this video. And between the old pair and the new one, between the old partner and the new one, a covalent bond is formed. So the ribonucleoside triphosphate, it bonds to the exposed complementary partner. And just like in the case of replication, the energy for this entire process comes from the cleavage of the additional phosphate groups to join them together. So theoretically speaking, this is the process of DNA transcription, and the sequence of DNA that is transcribed into RNA. The sequence of DNA transcribed into an RNA is called a gene. Gene for genetics, the same gene. And transcription, it occurs in the nucleus. And once it is made, the messenger RNA, so this RNA that we are talking about, the RNA that becomes this one, this messenger RNA, it moves from the nucleus into the cytoplasm. And this is where translation takes place. So transcription is in the nucleus, and the mRNA that is formed As a result of the transcription, it moves into the cytoplasm for the translation process. So, three main types of RNA are predominantly made. That is important before we go ahead and try to draw a diagram of this entire process. One is messenger RNA. It is nothing but a transit copy of the gene. That means a copy made for transport purposes only of the gene used to encode a polypeptide, which is the building block of proteins. So the messenger RNA is just a transit copy of the gene which is used to encode a polypeptide. Then we have transfer RNA. What is transfer RNA? It is a leaf-shaped sequence. That means it looks like this, a leaf-shaped sequence that carries an amino acid. So it has a number of sequences on them. And then finally, ribosomal RNA. This one is a primary component of ribosomes. So these three kinds of RNAs are going to be discussed and they will be used in the process. But before that, I'll give you an idea of how this entire process takes place. So let's suppose here was the original DNA strand, the double helix. Now this separates here like this, and one of these, it's used to form another pair. Now in DNA replication, what happens is a strand of DNA is formed, but in this case a strand of RNA will be formed. So let me just make it like this. This one is the messenger RNA. Eventually these two strands will combine together and this messenger RNA will be left alone. Now this messenger RNA, this strand is the transit copy, and this will move out of the nucleus and into the cytoplasm, and then it will take part in the process of translation, in which we will be using these two kinds of RNAs as well. So before going to translation, let us go ahead and describe the genetic code, which is often formed in terms of codons that are comprised of triplets of bases. So what is this genetic code? The genetic code is a set of rules by which information encoded in the mRNA, that means the messenger RNA, is converted into proteins. Proteins are nothing but sequences of amino acid by living cells. So this process does not take place in dead cells. So it's a set of rules by which information encoded in the messenger RNA or the mRNA is converted into proteins. Now codons are triplets of bases that encode a particular amino acid. There are 4 bases, so there are 64 codon combinations. How do we get this? 4 into 4 into 4. The order of the codons determines the sequence of amino acids for a particular protein. The coding region, it always starts with a start codon, which is usually a combination of A, U and G. Remember, we have U instead of T because we're talking about the messenger RNA, not the DNA. So every codon, it has a start codon, and there is a stop codon as well for the sequence, which is what we will be covering. So let me give you a few examples for the different kinds of codons that exist. These are a few examples. So if the codon has the pairs in this kind of series, then it's going to be phenylalanine. If there are codons in AUU, AUC or AUA, it will be isoleucine. So it's not possible to learn up all the kinds of proteins or the amino acids, but there are a few features that you need to know about the genetic code. Now, the genetic code is universal. That means every living thing uses the same code. There are few rare and minor exceptions, but otherwise it is universal. That means AUU will always code for isoleucine in all animals. So that means it is universal. It is degenerate. That means there are only 20 amino acids and 64 codons. That means more than one codon may code for the same amino acid. For example, here 3 are coding for the same one. In this case, 4 different combinations of codons, 4 codons can code for the same amino acids. That is why it is degenerate, and this allows for silent mutations whereby a change in the DNA sequence does not affect the polypeptide sequence. So let's suppose there was a mistake and instead of CCU, CCA got coded. Even then, the resulting amino acid is going to be the same. The polypeptide sequence is going to remain the same. So that kind of a mutation is known as a silent mutation, where a change is happening, but it is not resulting in an effect. So the next process that we need to do is the process of translation that leads to formation of polypeptides. So translation leading to formation of polypeptides. The translation is the process of protein synthesis, and in this what happens is the genetic information encoded in the mRNA, the messenger RNA, it is translated into a sequence of amino acids in a polypeptide chain. So what happens is the ribosomes, they bind to the messenger RNA in the cytoplasm. They move along the messenger RNA molecule. So the ribosome, it moves along, it like kind of slides along the messenger RNA molecule in a 5-3 direction till it reaches the start codon. So it clings on to the messenger RNA strand and keeps moving in the 5-3 direction till it reaches the start codon, which is always AUG. Now anticodons are translated. Anticodons are, let's say, transported through transfer RNA molecules. They align opposite. So on one side the messenger RNA is coming, on the other side the transfer RNA is coming, and they both combine in the ribosome, which I'll show with the help of a diagram later, but you can just make a note of the process that happens. So the anticodons on the transfer RNA, they align opposite to the codons according to the complementary base pairing. For example, USC is going to align opposite to AUG. So that is going to be the complementary pairing. Now each tRNA molecule, each transfer RNA carries a specific amino acid according to the genetic code, just like we did a few examples. Ribosomes, they catalyze the formation of peptide bonds. So they catalyze the formation of peptide bonds between the adjacent amino acids, and this is through a condensation reaction. The ribosome, it moves along the messenger RNA now. And in the result, a polypeptide chain is synthesized, and this movement takes place until it reaches the stop codon. At this point, the translation stops and the polypeptide chain that was created, that is released. So let me go ahead and draw a diagram so that it's much more clearer to you. So this is a diagram. This big structure that you can see, the purple colored one, this is the ribosome. Now this strand that you can see, this is the strand of messenger RNA, and this is one codon. Now this arrow, the blue colored arrows, they show movement. So the direction of the ribosome is this way, and it moves in a 5-3 direction on the strand. So if you go into detail and start drawing the base pairs, then the direction would be 5-3. Now these are the codons, they're moving, they're actually not moving, the ribosome is moving on them. And over here you can see this is the amino acid. So it enters the ribosome with this anticodon. The red ones are all anticodons. So what happens here is the peptide bond is formed. So this bond over here is the peptide bond. So this amino acid is going to come here. This H is going to form a peptide bond with G, and this GUG will combine with CAC, like over here. So these will come here. Similarly, G will bond with this one, R is going to bond with this one, and the polypeptide chain will come out in this direction. So I hope you're able to understand this, and the remaining will be known as the transfer RNA. So the remaining chains will go back, the empty ones without the peptide bond, without the amino acid. So this is the entire process of translation. So you can watch this video again to note down all the steps or to understand it a little more clearly. And this is the simple process of translation. Now what is the relationship between a gene and a polypeptide? That is very important. You know that gene is a sequence of DNA that encodes a polypeptide sequence. A gene sequence, it is converted into a polypeptide sequence by the process of transcription, that is by making an RNA transcript, and the process of translation, which is by synthesizing the polypeptide. So this is the process by which a gene sequence is converted into a polypeptide sequence. Now translation, it uses the RNA molecules, that means the transfer RNA molecules, and the ribosomes to join the amino acids into a chain of polypeptide according to the messenger RNA sequence, which means the codons. And since the genetic code is universal, it means all organisms show the same relationship between genes and polypeptides. That indicates that there is a common ancestry and lots of transgenic techniques can be employed. So some proteins, they may consist of a number of polypeptide chains and they need multiple genes. An example of that is hemoglobin. So some proteins may consist of a number of polypeptide chains, because usually one protein means one polypeptide chain. In some proteins like hemoglobin, hemoglobin has 4 polypeptide chains. Let's call them PP chains. And it has 4 polypeptide chains and they are encoded by 2 different genes. When a gene is mutated, it can lead to synthesis of a defective polypeptide, hence affecting the function of proteins. So gene mutation can lead to a defective polypeptide, that means the wrong one. That can lead to protein malfunctioning. This is what happens in lots of hereditary and gene-related diseases. So the rule that we follow is one gene, one polypeptide. That means in the majority of cases, apart from exceptions. So let's suppose we start with the DNA strand. Now I'm going to summarize everything. Let's suppose this is the DNA strand, this is the 5' end, this is the 3' end. Let's say some pairs are here. This is ACG, this is TTC, GAC, and TAG. Now through transcription, mRNA transcript will be created. This is going to be in the opposite direction because the strands are antiparallel, and this process is transcription. So over here you are going to have UAC complementary to ACG, AAG, CUG, because remember we are using U for A, now the RNA is being created, and finally GUC. So that is being created over here, not GUC, it's AUC over here. Now these are the different codons, and through the process of translation, a polypeptide will be created. The polypeptide does not have a direction like 5'3". So for example, we are going to have a polypeptide chain like this. This is just an example. Okay, so different shapes, and these are the component amino acids. So these are going to code like this. So I hope you'll be able to draw a better diagram. There are only two exceptions to this. So let me write them down here. One is genes coding for tRNA and the rRNA, that means the ribosomal RNA. They do not code for polypeptide sequences. They only the mRNA sequences code for polypeptides. So the tRNA and the ribosomal RNA, they do not have any role in creating new polypeptides. That means the tRNA has a little bit role, but tRNA does not create a polypeptide. You cannot place a transfer RNA or a ribosomal RNA in this place. It's only a messenger RNA. And the second exception is that a single gene can sometimes code for multi-polypeptides if splicing occurs. So that is a little detailed concept. So a single gene may code for multiple polypeptides if splicing occurs, which we'll be doing when we talk about advanced genetics and the advanced structure of DNA. So right now you do not need to know about that. And this is all that we'll be covering in this video. So I hope it was useful. What we covered was a very important process of DNA transcription and DNA translation. So basically formation of polypeptides from DNA. So thank you so much for watching this video.